myocardial cells when it comes to action potentials, which ions are involved when their permeabilities change, resting membrane potentials versus pacemaker potentials. All that stuff makes sense? Okay. All right. So we talked about heart rate modulation last time, how we can modify the heart rate. But let's look at the actual autorythmic rate. Assuming you sort of leave your heart alone, you're not dousing it with epinephrine, norepinephrine, or acetylcholine, what's your heart going to do all on its own? Well, it has its own rate. You can sort of think of it as like this baseline rate that it works from, and then we modify it from there, either speed it up or slow it down. And a typical healthy heart's autorythmic rate is kind of sort of around 70, 75 beats per minute. And what does it do to? Well, it's really due to the SA node, the sinoatrial node. Remember, that's where the electric signal begins in the heart. When that SA node, composed of autorythmic myocardial cells, when those cells depolarize, generate action potentials, that spreads through the whole heart and sort of initiates or kickstarts that cycle. And so really it's the SA node that sets the rate for the heart. Its natural rate is around, like I just mentioned, 70 beats per minute. It's around 70 beats per minute. So if your endocrine system and your nervous system were not influencing your heart, that's what your heart rate would be. So that SA node fires all on its own at around 70 beats per minute, or 70 times per minute, which would lead to 70 beats per minute. The AV node is also autorhythmic. Right? It has its own rate, and it's a little bit slower. It's around 50 beats per minute, or would lead to a heart rate of 50 beats per minute if it was the one in charge. The atrioventricular node. Remember, that's the one that's right between the atria and the ventricles. And the Purkinje fibers, they have their own rate, from around 25 to 40 beats per minute. So even slower than the AV node. Right, with all these different autorhythmic rates, though, why doesn't part of your heart beat at 70 beats per minute, and part at 50, and so on? Well, that's because the SA node, the fastest one, sets the rate. Right? So remember that these rates are what they would do if they were sort of left on their own devices. Right? The AV node would fire 50 times every minute unless something stimulates it earlier. And that something comes from the SA node. So there's an analogy we can think about trains. So let's say we've got our train with lots of cars here, and we've got 3 locomotives tied up to this train. The locomotive in the front represents the SA node. And let's say it likes to travel at 70 miles per hour. That's a fast train. The AV node, let's say, on its own, travels at 50 miles per hour, and the Purkinje fiber is at 30 miles per hour. But they're all hooked up to each other. Right? So the Purkinje fibers are not going to go slower than all the other cars. They're all going to go at the same rate. And they're going to go by the fastest rate. Because they're all attached to the SA node, as the SA node pulls at 70 miles per hour, all the other ones are going to go at 70 miles per hour. So that's sort of an analogy for how it works in your heart. If that SA node's firing at this higher rate, those action potentials spreading through the gap junctions are going to stimulate all those other cells to fire sooner than they would otherwise. Just like if it was doused with epinephrine, it's a similar idea. Right? You can speed up the rate that way. Everybody see how that works? All right, so let's say one of those locomotives jumps the track. That would be a nightmare. But let's say it happens. So the SA node gets shut down for some reason. The next highest rate is going to take over, or the next fastest locomotive, in this case the atrioventricular node. So now the heart is beating at 50 beats per minute, or the train's moving at 50 miles an hour. Because the fastest locomotive in this train is at 50 miles an hour. So it's pulling everything else at that same rate. It's not going faster because it's no longer connected to the SA node. So if something happens to your SA node and it stops firing, then your AV node's going to take over. And thank goodness that's there, because now your heart's going to keep beating and you'll stay alive. It's not going as fast, but it can be fast enough to keep you alive. Let's say the SA node's fine, but the AV node jumps the track. Well, that's a really big problem. Because your SA node's going to keep going at 70 miles an hour. your atria keep contracting at 70 beats per minute. The AV node no longer is working. So signals are not going through the AV node, which means the atrial signal is not passing down to the ventricles. So now the Purkinje fibers will take over at their rate. In this case, 30 miles an hour or 30 beats per minute. So now the SA node is firing and the atria are contracting at this higher rate of 70 beats per minute, and the ventricles are contracting at this slower rate of 30 beats per minute. And that's trouble. Right? They're getting out of sync, the ventricles aren't filling up properly, and the whole heart's going to get stressed, you're going to get these weird waves, and then eventually just stops. So you don't want that to happen. Right? Okay. How does everybody feel about these autorhythmic rates and how they influence each other? We see how the fastest pacemaker sets the pace? Okay, yeah?
My question is, why is, like, the Purkinje fibers are too slow to really be healthy for a human to live? Why are they at such a low rate? Is it like an artifact of early evolution, or?
I don't know, but my guess is that it is in part an artifact of ancestors, right? So if you look at other vertebrates' resting heart rates, they're often a lot lower. Like you look at fishes, their blood pressure is much lower, their heart rate's a lot lower. I'm not saying that that's left over from a fish heart. I'm not saying that at all, but it could be sort of a remnant of generations gone by. It could be. Yes?
What's the, I guess, the action of when this would happen?
When what would happen? When one of these cars jumps the track?
Yeah.
Some part of the heart doesn't form properly, those cells don't form properly and don't work. If some stress causes those cells to die, there's two ideas. What's that?
A whole bunch of stuff.
Yeah, I mean anything could go wrong in our bodies, right? And you think about it, it's pretty amazing that these complex things work as well as they do. Yeah.
Is that where the whole pacemaker comes into play? Like they hook it up to one of these nodes?
So it can, right? So if you're getting, problems with these, then like your SA node is no longer working, then you can have a pacemaker, an artificial SA node with a little electrode that goes into your atria and zaps it when you want it to go. Right? And so that's what it could have there. Or if your AV node is knocked out, you can have something that can join those two or takes over for it and accommodates for this lack of cells. So yeah, pacemakers can help with this. Anything else? Yeah.
But what happens if one of these things happens? Like if I'm sitting here and one of these two things happens to my heart just like right now, is it something that I'm sure I would notice, but is that considered like yourself having a heart attack? Like what
not necessarily a heart attack. Oh gosh, there's a heart attack and cardiac arrest. One is with the heart attack is the myocardial infarction, right? Is that the one where part of the heart tissue dies, or?
Yeah.
Yes.
And then the cardiac arrest is when blood flow. There was something with blood flow. I can't remember the details, but so this doesn't necessarily immediately lead to one of those two things. Right? There's all these different things that can happen to your heart to cause it to stop. And I believe if your SA node stops, you might notice a difference, get tired faster and strained. I don't know that it necessarily leads to your heart stopping, where this one would lead to your heart stopping because they've got these two different rates and it just doesn't work together and puts too much stress on the heart. So it kind of depends on what goes wrong and to what extent. Anything else? All right. So here we've got our heart. We saw this before. The yellow represents what part is being depolarized. Starting at the SA node, goes through the atria, goes down that interventricular septum to the apex, and then comes back up the Purkinje fibers, and then we repolarize the ventricles. And we can go ahead and map out that electrical activity. If we use surface electrodes to record the electrical activity, and that is called the electrocardiogram, which you guys are already familiar with, so this is going to be a quick review. I think we mentioned it last time also. Remember, this is a recording of the heart electrical activity. That's what the ECG is, or electrocardiogram. EKGs are exact same thing. Cardio spelled with a K. I think that has to do with the language of origin that this device or this method was originated from. All right, so let's just go back over the ECG waves real quick. I know you guys all have this straight, but just to make sure, remember the P wave is due to the atria depolarizing. So that's the first wave you find in the ECG. So when the atria depolarize, that's recorded as a P wave. The QRS complex is due to the ventricles depolarizing, or ventricular depolarization. Timing-wise, this is also where you get atrial repolarization. Right? So the atria are done depolarizing, they're done contracting, and they repolarize at this point. And then the third part is the T wave, which is ventricular repolarization, or the ventricle repolarizing. Okay, everybody got that? Okay.
For blood pressure, is it happening this way? For example, 120 over 80, is it between these two, like T wave to P wave, or?
What contributes to those two numbers you get in blood pressure? So those two numbers have to do with the pressure that results from your ventricles squirting blood into the arteries. And we'll talk about that a little bit later, why those are two different numbers. The blood pressure really swings between those two extremes. And that has to do with the blood pressure in your arteries when the ventricles contract and then between contraction. Right? Between contraction, the pressure drops, and so it gives you that lower number. So where is all that pressure coming from? It's between the QRS complex and the T wave. The QRS complex is when you stimulate the ventricles to contract, and this time between the two is when the actual contraction occurs. And that's what's generating all this pressure of squirting the blood into the arteries, which leads to pressure in the arteries. Does that work?
And 80, the two number, one of this 80, one of this 120.
Yeah, your blood pressure could be 120 over 80, yeah.
Which of this 80, which of them covered for 120?
Which is which?
Which of them is covered for 80, which of them covered for 120?
Which one's
Like,
covered?
which part of the heart for the access, which we...
Well, those pressures are always usually measured in your arm, actually, on an artery going through your arm. But those pressures directly relate to what came from the ventricle, the left ventricle. Because that's what pumps the blood out into the systemic circuit, including your arm. So we're going to talk about that swing in pressure a little bit later and the pulse that you get from it. Okay? Anything else for now? All right, so here's an ECG. We've got the P wave due to atrial depolarization that leads to atrial contraction. Then we get the QRS complex. When the ventricles depolarize and initiate ventricular contraction, we also get atrial repolarization here. So by the time we get to the T wave, the ventricles are done contracting and the ventricles repolarize. And then this longer flat sections where the heart fills back up, and we do it all over again. Atria contract, ventricles contract, refill. Okay, questions about that? All right. So the ECG, remember, that's recorded from surface electrodes. So you've got electrodes on either side of your heart, which is very different than having one electrode inside a cell and one electrode on the outside of the cell. When we talked about action potentials last time, in contractile myocardial cells and autorhythmic myocardial cells, those electrical signals that we were talking about were from transmembrane electrodes, electrodes that were across the cell membrane. So that's very different than what you get with an ECG. But I want to correlate the two so you can kind of put it together in your head. So we've got time on the horizontal axis, and we've got potentials on the vertical axis. Right? This bottom one is from a transmembrane reading across the cell membrane. Is this from an autorhythmic cell or a contractile cell? Why do you say that?
Why is it plateau?
That's right, so the contractile cells have the plateau. What's another indicator?
It doesn't, well, it has a resting potential.
Yeah, you've got a resting potential down here. Autorythmic cells don't have that. Nice job. Okay, so this graph at the bottom, this represents the action potential from one individual cell. Keep in mind, when your heart beats, it's lots and lots of cells all beating at the same time. They'll be slightly out of sync of each other, and that's what all these signals are coming from, all of these cells together. Right? Remember, when we use these surface electrodes, it's recording all of the electrical changes between those two electrodes, anything that happens in there. All right, so we've got this resting potential here in this contractile cell. This will represent a contractile cell from the ventricle. And notice during atrial contraction, nothing's happening in this cell down in the ventricle. But once we get to the QRS complex, you can see that that cell depolarizes, which starts its action potential. And then it starts to repolarize, and then you get the plateau phase. And that plateau phase takes it all the way over here, and then we finally finish repolarization, which matches up with the T wave. So this slope here represents, or correlates with that T wave. That's where that T wave is coming from. It's from all of those cells in the ventricles repolarizing. So notice that this entire time it's going through the action potential, that's spanning between the QRS complex and the T wave. Right? And that also correlates with the contraction of that cell. It contracts here, and then it finishes relaxing over here. And we talked a little bit about that last time, the benefit of that plateau phase. Right? It's keeping the heart from summing up contractions, right? Keeping it from going into tetany. All right, so we've gone through the whole ECG. You can see that it's a cycle that keeps repeating, it keeps going and going, right? Once you've finished repolarizing the ventricles, you fill back up and you start all over again. It keeps going. And really what this leads us into is the cardiac cycle. And that's what I want to focus on now, the cardiac cycle. And maybe we will get through it all. We should leave it on the exam.
We'll see.
We'll see.
Don't give you the
It's a
time.
parent thing to say. Oh, we'll see. We'll talk about that later. And hopefully after you forget about it. All right, cardiac cycle. So the cardiac cycle is going through sort of one complete cycle of contracting and relaxing. Right? We're going to fill up the heart, squirt all that blood out, and then fill it up again. So what I want to do is go through that whole cycle and look at lots of different aspects to that cycle. Okay, so for one, we'll look at contraction. Contraction of the atria and contraction of the ventricles. But of course, we can't just call it contraction. We have to introduce a new word, so you have more to memorize. So we're going to call that systole. All right, so systole refers to contraction. Right, so you'll hear systole and systolic, same thing. So you can talk about atrial systole, that's when the atria contract, and ventricular systole, when the ventricles contract. Then we're also going to take a look at relaxation, when the chambers relax, and this is called diastole. Again, you can have atrial diastole and ventricular diastole. So look at when, during the cycles, the two different types of chambers are going through systole and diastole. We will also look at the electrical activity. We've already done that, actually. You guys know that already. We'll take a look at the pressures involved. So as you go through a complete cardiac cycle, what are the pressures like in the atria, in the ventricles? We will look at the sounds produced. All right, so typically, hearts through one cardiac cycle generate two different sounds, often referred to as the lub and the dub. Lub-dub, right? And so we'll take a look and see what those sounds really are, right? When those occur. So sounds that we usually hear with the stethoscope correlate to the valves in your heart closing. So when the valves close, they make a noise. Okay? We'll also take a look at the volume of the chambers. Right? When are the chambers getting larger, filling up, and when are they emptying? We'll take a look at that as well throughout the cardiac cycle. Everybody got this down? All right. So we're going to break up the cardiac cycle into 5 phases. I think your book uses 4 phases. Some books use 4, some use 5. We're going to use 5. The first phase is late diastole. And what does diastole mean? Relaxation. All right, so the whole heart's going to be relaxed during late diastole. Then we're going to take a look at second phase, atrial systole, when the atria are contracting. So some places you're going to read it where phase 1 and 2 are sort of lumped into one bigger phase. But there's a clear difference of what's going on in the heart between late diastole and when the atria start to contract, so I like to emphasize that. The third one, this is a fun one. Isovolumetric ventricular contraction. Seems long and awkward, but it's a very useful name because it tells you everything you need to know. It tells you what's going on there. All right, so it's clearly when the ventricles are contracting. And the word isovolumetric, what does iso mean? Same. So the volume of the ventricles will be constant during this phase while the ventricles are contracting, which at first sounds kind of weird. Why would it contract and not change volume? We will see. The fourth phase is ventricular ejection. So this is when blood is ejected from the ventricles. And the last phase, it's another isovolumetric phase. This one's isovolumetric ventricular relaxation. So the ventricles are relaxing.
The reason why the first is it's diastole, but then number 5 refers back to relaxation.
Just to keep it confusing,
Okay.
I don't know why it's done that way. I've always seen phase 3 and 4 or 3 and 5 written that way with contraction relaxation, and I don't know why. Just to make sure you're comfortable with both words, maybe. Okay. All right. So what I want to do is we're going to go through the cardiac cycle a number of times. And we're going to try and go through them different ways. So we're going to go with the same material in different ways to try and sort of highlight different parts, get it stuck in your head. So now what I want to do is I want to go through it, and we'll first go ahead and sort of track the ventricles. And then we'll go through and look at so we'll look at the atria first, then the ventricles, and we'll sort of tack things on as we go along. All right, let's go ahead and look at this first figure here. So, when we first start looking at these figures, they're going to look really confusing. I'm going to try and break them down into sort of smaller pieces, and then at the end, kind of put it all together, and hopefully it makes perfect sense. Okay, so for all of these, we've got time on the horizontal axis. Right? So this is going through, in this case, 2 cardiac cycles, and well, 1.5, 1 and then some. The bottom here, we have the volume of the atria. Right? It's what. Sorry. This is the volume of the ventricles. This is the volume of the ventricles. So we have down here, and we've got the state of the valves up here, showing when they're open and when they're closed. So the white portions represent when the valves are open, the blue portion when the valves are closed. And then on top we have the phase numbers. Remember, our phase 1 and 2 is really the phase 1 here. So we break up phase 1 into 2 separate phases. And I'll try to highlight that as we go through. Okay, so for this one, we'll sort of concentrate on the volume of the ventricles. So our first phase was late diastole. So all the chambers are relaxed. Nothing is contracting, nothing's stimulated in the heart. Blood's coming back from the veins and pouring into the heart. We're filling the heart. And so that corresponds to this part right here. It also corresponds to this part. This is just in the next cycle. So this is during late diastole. The heart is filling up. While it's filling up, Blood's pouring in from the veins into which chambers?
Atria.
The atria. So the atria are filling up during this time. But also during late diastole, as you can see from this figure, the ventricles are filling up. So some of the blood that pours into the atria pours through into the ventricles. So during late diastole, all 4 chambers are getting filled with blood. If that's happening, if blood is pouring into the ventricles, what must be true about the AV valves? They've got to be open. The only way you're going to get blood into... something stuck in my throat. I'll be okay, don't worry. The only way to get blood into the ventricles is through an open AV valve. And we can see that the AV valves are open during late diastole. They're open now. The semilunar valves. What state are they in? Well, this chart tells you they're closed. And that makes sense because blood is not leaving the ventricles. Instead, blood's entering into the ventricles. And we will see this throughout the cardiac cycle that there is no time when all 4 valves are open at the same time. So, what are the options? Blood can come into the ventricles, AV valves are open. Blood can leave the ventricles when the semilunar valves are open. Or, no blood's coming in and no blood is leaving. And that's when all the valves are closed. So those are our 3 options. There's no time when all the valves are open. Okay, so that's late diastole. Now we're going to go into our phase number 2, so that would be atrial systole. The atria contract. You get a P wave, the atria squeeze down, they squirt their contents into the ventricles. So sure, the ventricles were filling up before, but they weren't completely full yet. So when the atria contract, you get this little surge in volume in the ventricles. You're going to push even a little more blood into those ventricles. You top them off. So that's really what happens during atrial systole is you top off the ventricles. And that's why we see this little surge in volume in the ventricles right here. While that happens, the AV valves must be open. We're still putting blood into the ventricles. And the semilunar valves are still closed. Blood is not leaving yet. So far, so good? I know it kind of seems painful how slow we're going, but I'm trying to do it in little tiny chunks to have it make sense. All right, let's go on to our 3rd phase, which is isovolumetric ventricular contraction. It's a long name, but it's a short duration. This little bit right here represents our phase 3. Notice that the volume of the ventricles does not change during isovolumetric ventricular contraction. As the name suggests, it's a constant volume. The volume doesn't change. So if the volume doesn't change, it's no longer getting larger, so blood's not coming into the ventricles. So what must be true about the AV valves? They must be closed, right? They close. And blood's not leaving the ventricles yet. And so what must be true about the semilunar valves? They're also closed, like it shows here. So you can see they're both in that blue state, so they're both closed. So the ventricles, they started contracting. They're starting to squeeze down in their contents. Well, what caused the AV valves to close?
Pressure.
It's always pressure with valves, right? So that pressure shot up in the ventricles to shut those AV valves. We'll see that more with pressure charts in the next one. All right, so that's our phase 3. Now we go on to our phase 4, ventricular ejection. And that is the phase 3 on this figure right here. So the ventricles continue to contract, continue to squeeze down on their contents. And we can see that blood is now leaving the ventricles. It's squirting out into the arteries. The only way for that to happen is the semilunar valves must have opened, and we can see that on the chart up here. Semilunar valves are now open. So those ventricles are squeezing down hard enough now to blow those valves open and let blood squirt out into the arteries. The ventricles are ejecting their contents. Alright, that keeps going until we get down to the bottom here when we go into our last phase, isovolumetric ventricular relaxation. So now the ventricles are going to start relaxing. And as they relax, that back pressure in the arteries is going to slam those semilunar valve shut. Semilunar valve is now closed. And at that point, no more blood will leave the ventricles. The door to leave the ventricles is closed now. And notice that it's an isovolumetric phase. The volume is not changing. So not only is blood not leaving the ventricles, there isn't any blood coming in yet. So those AV valves are still closed from before. Right? Way back here, we closed the AV valves. Well, they're still closed all the way through this second isovolumetric phase. All the valves are shut, no blood's coming in, no blood's going out. The volume is constant in the ventricles. Questions about that? Is this making sense? Is it too much? Too little? Just right. Okay. Let's look at another one. So here we're sort of focusing on the volume of the ventricles. Now let's talk about pressure. We're going to focus on pressure. And we can correlate it to the volumes and to the state of the valve. Alright, so here we have the pressure of the ventricles in green. So this curve right here represents ventricular pressure, and this purple, we're going to call it purple. This purple line represents the pressure of the atria. Okay. As we go through, by looking at these two pressure lines, that alone can tell us what the state is of the different valves. Every time those two pressure lines cross between the atria and the ventricles, you're going to get a change in your AV valves. Remember, they're driven by pressure gradients. It's all based off of which side has the higher pressure. Those lines cross, that means you're switching who has the higher pressure. And we'll have to compare the ventricular pressure to the pressure, let's say, in the aorta to figure out the semilunar valve. We'll do that in the next slide. Okay? Alright, so let's start with late diastole. So during late diastole, all the chambers are filling up. Right? So the AV valve must be open, AV valves, to let blood into the ventricles. And the semilunar valves must be closed because blood's not leaving the ventricles. And if you look at the pressures in these two chambers, this is supposed to represent the left side of the heart, but the same thing's happening on the right side of the heart at the same time. You can see that atrial pressure is higher than ventricular pressure. It doesn't have to be higher by much, it just has to be higher. And that keeps those AV valves open so blood passes through the atria into the ventricles. And that's what we get here while the ventricles and the atria are filling up. AV valves are open, semilunar valves are shut. Yes?
Can you repeat that again? You said atrial pressure is higher than atrial...
When the atrial pressure is higher than the ventricular pressure, the AV valves are open. Anything else for now? Yeah.
What phase is this?
We were talking about late diastole.
Okay.
So the heart's filling up. We haven't started contracting yet. Okay? All right, so let's go on to atrial.